Imagine trying to learn a completely new language from scratch.
Okay.
But the textbook you're handed is just fundamentally broken. Like 90% of the words on every single page are just random filler.
Absolute gibberish.
Exactly. You're reading along, trying to figure out the grammar, and for every 10 words you read, 9 of them mean absolutely nothing. Only, one single word actually moves the plot forward.
Your task in that scenario is to somehow deduce the entire complex storyline of the book using only that sparse 10% of meaningful text.
Right.
All while trying not to get completely overwhelmed by the sheer volume of noise surrounding it.
It sounds like an impossible, maddening task.
Yeah.
But that scenario is exactly the landscape artificial intelligence faces right now when it tries to read our genetic code. Welcome to today's custom-tailored deep dive.
Thanks for having me.
Our mission today for you listening is to dig into a truly groundbreaking research paper. It was published in Nature Methods, and it introduces something called Orthrus.
Yeah, Orthrus. It's a revolutionary foundation model for mature RNA.
And the reason we are focusing on this specific paper today is because it fundamentally challenges the dominant philosophy in the tech world right now.
It really does. The whole industry is obsessed with this idea that to build a better AI, you just need a bigger computer.
Right, exactly. You buy more graphics cards, you build massive server farms, you feed the machine more data, and eventually it just magically gets smarter.
The industry calls it the scale is all you need approach.
Scale is all you need, but biology, as we're going to see, operates on a completely different playbook.
Oh, absolutely. What we are exploring today is proof that in the biological domain, working smarter completely destroys the strategy of just working harder.
Working smarter, meaning scientists have figured out how to give AI the ultimate biological cheat sheet, right?
Yeah.
Using billions of years of evolutionary history as a guide.
Yeah, that's exactly what they did.
So to understand why Orthrus is such a massive paradigm shift, we have to look at why current AI models are failing so spectacularly at reading genetic code. I mean, we have these incredible natural language processing algorithms, right? The tech behind things like ChatGPT.
Right, the large language models.
Yeah, they can write poetry, they can write software. Why are they hitting a brick wall when we point them at the human genome?
Well, it comes back to your opening metaphor about the book of gibberish. Most current genomic foundation models are built on techniques like next token prediction or masked language modeling.
Okay, let's define those terms for someone who doesn't build AI for a living. What is a token and how does that prediction work?
Sure. So think of a token as a single piece of a sequence. In English, a token is usually a word or maybe a syllable.
Cool.
In genomics, a token is a biological letter, so A, C, G, or U in the case of RNA. Now, masked language modeling is basically like a fill-in-the-blank test.
Like Mad Libs.
Sort of, yeah. The AI is given a sequence, one letter is hidden or masked, and the AI has to guess what that hidden letter is based on the surrounding context.
That makes sense for human language. Like if I say to you, the dog chased and I hide the next word, the AI can reliably guess cat or ball.
Right.
Because almost every word in a human sentence is there for a structural or narrative reason.
Exactly. But the human genome doesn't play by those rules. Approximately 90% of the human genome lacks any evidence of what evolutionary biologists call negative selection.
Okay, I have to push back on that a little bit. When we say 90% of the genome is just noise or junk, are biologists absolutely certain about that? Or is it just stuff we haven't figured out how to read yet?
No, it's a very fair question. And the term jump DNA is definitely controversial, but the concept of negative selection gives us a mathematical way to look at it.
How so?
Well, negative selection is basically evolution's quality control department. Imagine a mutation occurs in a highly critical part of our DNA, say the blueprint for a vital heart protein.
Okay, that sounds bad.
It is. That mutation is likely harmful. The organism will be less fit, it might not survive, and crucially, it won't pass on that mutated trait.
So evolution actively selects against that change, meaning over millions of years, that critical sequence stays mostly the exact same because changing it breaks the machine.
You've got it. But exactly the opposite happens in that other 90%. Mutations happen there constantly, but they don't seem to significantly impact the organism's survival or fitness.
Oh, I see.
So evolution doesn't step in to preserve those areas, they just drift and change randomly over time. From the perspective of figuring out what actually makes a cell function, that highly mutable 90% is just low information noise.
So if an AI is using next token prediction, if its entire goal in life is just to guess the next letter, and 90% of the letters it's looking at are just random evolutionary drift.
It's going to spend all its computing power trying to find patterns in the static.
Wow. It's trying to find grammar where there is no grammar.
Exactly. The AI actually gets really good at reconstructing non-informative, useless tokens. It memorizes the static on the television screen instead of paying attention to the broadcast.
And the tech industry's reaction has been to just throw more processing power at the static.
Pretty much, yeah.
They scale these models up to billions of parameters, which are essentially the artificial synapses in the AI's brain. But this paper points out that adding more parameters to predict useless genetic static yields drastically diminishing returns.
Because if the AI is being rewarded for memorizing noise, we cannot expect it to predict the complex dynamic behavior of a living cell.
Right.
So the creators of Orthrus realized they couldn't just feed the AI raw linear text and hope it figured things out. They needed to give the AI a strong biological inductive bias.
Okay, inductive bias.
Yeah.
Which means giving the AI a set of assumptions or rules before it even starts looking at the data, right?
Yeah.
Pointing it in the right direction.
Yes. They had to teach the model how to group the genetic letters by their actual biological meaning, rather than just reading them blindly from left to right. And to do that, they used a technique called contrastive learning.
Contrastive learning is such an elegant concept. You don't ask the AI to guess the next word. You show the AI two sequences, and you say, these two things do the same job. Pull them together in your mathematical map, maximize their similarity.
Exactly.
And then you show it two unrelated sequences and say, these are different, push them apart.
A good way to visualize it is teaching a child how to sort objects. You show them a picture of a sedan and a picture of a massive pickup truck. They look entirely different.
Right, totally different shapes.
But you tell the child, these are both vehicles that drive on roads, group them together. Then you show them a bicycle and say, that requires pedaling, it's different, push it away.
So the AI learns the underlying concept of an engine-powered vehicle, rather than getting distracted by the paint color or the size of the tires.
Precisely.
Yeah.
And the genius of the Orthrus paper is what they chose for those related sequences.
Yeah, because how do you find two RNA transcripts that have entirely different genetic spelling, but do the exact same job in the body?
They use two brilliant biological cheat sheets. The first one is alternative splicing.
Right. Alternative splicing is a biological mechanism where a single gene can be cut up and pasted together in different configurations to produce different mature mRNA isoforms.
Yes, isoforms.
Think of it like a director shooting a movie. They have all this raw footage that's the gene. They can edit that footage together in different ways to create a theatrical cut, a director's cut, or an extended edition.
That's a perfect analogy. And the researchers took these different RNA cuts from the same gene across 10 different organisms. So humans, mice, chickens.
Wow.
And even though these sequences literally have different genetic lettering because they are edited differently, they belong to the same functional family.
So by feeding those pairs to the AI and saying, pull these together,
Yeah.
the AI is forced to figure out what they have in common.
Exactly. It learns to ignore the parts that change the noise and isolate the specific sequences that actually dictate the function.
Let's do a quick recap for the listener before we move on, because this is dense stuff.
Sure.
So far, we've learned that standard AI tries to read genetics like an English book and gets lost in the junk DNA. To fix this, Orthrus uses contrastive learning to group things by function rather than just spelling. And the first way they did that was by comparing different edits of the same gene, the alternative splicing.
Spot on.
Which brings us to the second cheat sheet they used, and this is the one that really blows my mind. They used evolutionary speciation, specifically tapping into something called the Zoonomia project.
The Zoonomia project is incredible. It's a massive data set mapping orthologs. Orthologous genes are genes found in different species that evolved from a common ancestral gene.
Okay.
They looked at transcripts across over 400 different mammalian species.
Wait, over 400. So we are talking about comparing the genetic code of a human to a bat, to a whale, to a naked mole rat.
All of them, yeah. Bringing it back to negative selection, Evolution aggressively preserves the sequences that are critical for survival across all those species.
Right, if it ain't broke, don't fix it.
Exactly. If a specific RNA sequence motif does something essential, it will look remarkably similar in a bat and a human, even separated by tens of millions of years of evolution.
That is so smart. They didn't have human scientists sit there in a lab manually tagging which parts of the RNA are important. They just use 400 mammalian species and millions of years of life on Earth as a giant automated filter.
The ultimate filter.
They literally just told the AI to group the bat sequence and the human sequence together.
And the results of doing that were definitive. The researchers performed ablation studies where they systematically remove one component of the training data at a time to see what breaks.
Like pulling wires out of an engine to see which one stops the car.
Yes. And when they removed that evolutionary ortholog data, the model's ability to predict RNA function plummeted. That contrastive learning objective, pulling together evolutionary cousins, is the absolute core of why Orthrus works.
Okay, so we have this brilliant training method that slices through the genetic noise. But having the right data is only half the battle, isn't it?
It is.
You also need a software engine capable of physically processing these massive biological sequences without the computer just crashing.
And mature mRNA sequences are not short sentences. They can be incredibly long, sometimes stretching to 12,000 nucleotides or biological letters.
Which brings us to a major technical problem with the current king of artificial intelligence. The transformer architecture. ChatGPT, Claude, Gemini. They're almost all built on transformers.
They are.
And while transformers are incredibly powerful, they have a massive vulnerability when applied to long biological sequences. Their memory requirement scales quadratically.
Right. Let's break down what quadratic scaling actually means in this context.
Please do.
A transformer works by looking at every single token in a sequence simultaneously and calculating how it relates to every other token.
I like to imagine a detective standing in front of a corkboard with pictures of suspects.
Okay, I like where this is going.
If there are 10 suspects, the detective has to draw a red string connecting every suspect to every other suspect to see how they relate. It's a lot of string, but it's manageable, right?
But if the sequence gets 10 times longer, say, 100 suspects, the amount of red string required doesn't just get 10 times bigger, it gets 100 times bigger.
Oh, wow.
If you jump to a sequence of 10,000 biological letters, the computer has to calculate hundreds of millions of individual connections all at once. The memory requirement just explodes.
It's incredibly inefficient. It eats up all the computing power just trying to hold the whole sequence in its head at the same time. So the Orthrus team completely abandons the transformer.
They did.
They build their model using a newer architecture called a Mamba state space model. Mamba?
Yeah, Mamba solves that memory explosion. Instead of scaling quadratically, Mamba scales linearly. So if a sequence is 10 times longer, it only requires 10 times the memory.
How does it actually achieve that mechanically? If it's not looking at everything all at once like a transformer, what is it doing?
Mamba processes information sequentially, but it maintains a hidden state, which is basically a running mathematical summary of what it has seen so far.
A running summary.
Think of reading a dense textbook. A transformer tries to memorize the exact location of every single word on every page simultaneously. Mamba is more like a student reading the book with a highlighter.
Oh, I like that.
As it reads, it updates its running summary with the important concepts, and it actively discards the filler words from its memory entirely.
So it has a mechanism to filter as it goes. I think the paper calls it selectivity. It looks at an incoming token and mathematically decides, is this relevant to my running summary? If yes, keep it. If no, throw it out immediately.
That selectivity naturally filters out the 90% of genetic noise we discussed earlier. Furthermore, Mamba excels at something called variable motif spacing.
What is variable motif spacing? And what is a motif, actually, just to be clear?
A motif is a specific recurring pattern in the DNA or RNA that has a biological function, like a sequence that tells a gene to turn on or off.
Okay.
Got it. This is crucial for biology. In human language, grammar dictates that words are spaced somewhat predictably. An adjective usually sits right next to a noun, but regulatory patterns in genetics, those little motifs don't follow rigid spacing rules.
So they can move around.
A lot. Two connected motifs could be right next to each other, or they could be separated by 3,000 random letters of junk DNA.
And a transformer struggles when important context is separated by thousands of useless tokens. But because Mamba is selectively ignoring the noise as it scans, it can effortlessly bridge that vast variable distance.
Exactly. It remembers the first important motif in its hidden state, ignores the 3,000 letters of static, and instantly connects it when it finds the second important motif.
That is wild.
The architecture perfectly mirrors the biological necessity of finding sparse signals in a sea of noise.
Let's do another quick recap for the listener. We've got the evolution-powered dataset teaching the AI what matters. And now we have this highly efficient Mamba engine that reads like a student with a highlighter, actively filtering out the junk and connecting the important dots.
That's a great summary.
So the ultimate test is putting Orthrus into the ring against the biggest, most expensive AI models in the world. How did it do?
Well, the researchers evaluated Orthrus on predicting incredibly complex mRNA properties. For instance, mRNA half-life.
Right, which measures how long an RNA molecule survives in a cell before it naturally degrades. A metric that is notoriously difficult to predict, but is absolutely foundational for designing things like mRNA vaccines.
Exactly. They also tested it on mean ribosome load, which determines how efficiently the RNA is translated into an actual protein.
Okay.
And subcellular localization, predicting exactly where in the cell the RNA will physically travel to do its job.
They matched Orthrus against several massive models, but the Goliath in this scenario is Evo2. Evo2 is a state-of-the-art genomic foundation model built with 7 billion parameters.
7 billion.
Right. And for context, the primary Orthrus model used in these tests has only 10.1 million parameters.
I really want to emphasize that for you listening. In the AI world, we are conditioned to believe bigger is always better. Evo2 has 7 billion parameters. Orthrus has 10 million.
It's tiny in comparison.
Orthrus is roughly 700 times smaller. I would naturally assume the 7 billion parameter model is going to crush the smaller one through sheer brute force.
In almost any other domain, it would. Yet across 7 different predictive tasks, this tiny Orthrus model either matched or completely outperformed the 7 billion parameter Evo2. Beating a model 700 times your size is staggering.
Yeah.
But the paper reveals another superpower that might be even more important for practical science. And that's few-shot learning.
Few-shot learning is a game changer. In traditional machine learning, training an AI to predict something like human mRNA half-life requires massive data sets.
Right.
You need to physically run experiments in a lab, label thousands or millions of RNA sequences with their exact half-life, and feed that massive library into the computer so it can learn the pattern.
And generating that physical data takes years and costs millions of dollars.
But because Orthrus already understands the foundational language of RNA because it spent its pre-training learning from 400 mammalian species, it barely needs any new experimental data to learn a specific task.
It's already primed.
Exactly. When researchers tasked Orthrus with predicting human mRNA half-life, it achieved 71% of its maximum possible accuracy using just 30 labeled training examples.
30, not 30,000, 30 data points.
Just 30.
If you are a researcher listening to this or just following the biotech space, the implications of that are wild. It means a small lab with limited funding can take a massive computational shortcut. They don't need a supercomputer or a $1 million dataset to test a hypothesis. They can do state-of-the-art computational biology from a laptop.
It completely democratizes the entire field. But predicting general properties like half-life is still just scratching the surface. The holy grail of RNA biology is taking a flat, one-dimensional string of genetic text and decoding its true three-dimensional biological function.
We mentioned alternative splicing earlier, one gene producing different RNA isoforms. The real-world consequence of that splicing is that a single gene can produce completely different proteins that do entirely different things in the body.
Right.
And historically, looking at 2 RNA sequences that are almost identical in their lettering and trying to guess which one does what has been near impossible for computers.
Yeah, it's been a huge bottleneck. But the paper proves that Orthrus has essentially developed a mathematical intuition for this. When Orthrus processes a sequence, it maps it into its latent space.
Let's visualize latent space for a second. Imagine a massive, multidimensional topographical map inside the AI's brain.
Okay.
When it reads a sequence, it drops a pin on that map. The rule is simple. Sequences that behave similarly in the real world get pinned close together. Sequences that have different functions get pushed far apart. Is that right?
That's a perfect way to picture it. And the researchers found that Orthrus's latent space actually clusters these isoforms based on their distinct biological functions, even when their raw genetic sequences overlap so much that they look identical to a standard AI.
The paper highlights a stunning case study regarding the OAS1 gene. This gene is heavily involved in our immune response to viruses.
Yes, the OAS1 gene produces two major isoforms, known as P42 and P46. Now, if you just look at the raw sequence, they share the vast majority of their genetic code.
So they look the same on paper.
Almost identical, but functionally they are worlds apart. The P42 isoform sits freely in the cytosol, the liquid inside the cell. It's essentially passive. It does absolutely nothing to stop viral replication.
It's just floating around on standby.
Right. The P46 isoform, however, is structurally slightly different. It receives a lipid group, which acts like a biochemical anchor.
An anchor?
Yeah. That anchor allows P46 to physically attach itself to a structure inside the cell called the Golgi apparatus. And from that highly strategic anchored position, P46 actively blocks the replication of viruses, including the virus that causes COVID-19.
So we have two outputs from the exact same gene. One does nothing. The other actively, physically anchors itself to a cellular structure to fight off a pandemic virus.
Exactly.
When Orthrus looked at the text sequences for P42 and P46, what did it do?
It recognized the hidden functional divergence immediately. In that mathematical map, its latent space, Orthrus plotted the P42 isoforms entirely separate from the P46 isoforms. It pushed them apart because it understood purely from the text that the tiny differences in their sequences translated to entirely different physical behaviors in a 3D cell.
The researchers even double-checked the AI's math using AlphaFold 3, which is the system that predicts 3D protein structures, right?
It did, yeah.
They found genes where the RNA sequences overlapped by 93%. A transformer model would look at that and say, these are the same thing. But Orthrus pushed them far apart. And when the researchers folded those sequences in AlphaFold 3, the resulting 3D physical structures were completely distinct. Orthrus was right.
Orthrus isn't just doing advanced text pattern matching. By correlating those incredibly subtle sequence differences with functional protein domains, it is inferring three-dimensional real-world consequences straight from a one-dimensional string of letters.
It's looking at the book of gibberish, filtering out the static, and seeing the matrix underneath.
That's exactly what it's doing.
A 10 million parameter model cracking the code of biological function by standing on the shoulders of mammalian history. It's incredible.
It really is a massive leap forward.
Well, this brings us to a final provocative thought to leave you with today. We've just explored how scientists use the sprawling, chaotic branches of Earth's mammalian evolution to train a highly efficient AI to read human RNA. I want you to mull this over. What other modern, impossibly complex data problems are we currently trying to solve with brute force computing, where the solution might already exist in nature?
That's the real question.
If we stop treating the history of natural evolution just as a subject to be studied in a biology textbook and start treating it as the ultimate pre-computed training algorithm, what other ancient cheat sheets are waiting to be decoded?
Nature has been running its own trillion-parameter experiments for 4 billion years. We are finally learning how to read the log files.
Thanks for joining us on this deep dive into Orthrus. Keep questioning the noise, and we'll see you next time.